Okay, nice to meet you all. My name's John Ellis. I'm from the University of Plymouth. I do like to explain when I give talks to beekeepers that I'm not a beekeeper myself. My background, I originally worked on bumblebees, and I use molecular techniques to study natural populations of plants and animals in a field called molecular ecology. As I said, I worked for a long time on bumblebees. More recently, with my colleague Mary Knight at Plymouth, we moved into working on honeybee systems. And I'm here to talk to you about our project, Shall Summer's Honey Breath Hold Out, the status of free-living honeybees in the southwest. It's a rather Shakespearean title because I stole a line from Shakespeare's sonnet 65. But when Ollie started on the project, he quite rightly pointed out to me that was rather a pretentious and long-winded title, and we'd be better off having something simpler and more straightforward. So it's now called Free Living Honeybees of the Southwest. And actually later on I'd like to get your feedback on our project and how we can get the public engaged. And one thing that we've been looking for is a catchy title to catch public attention. And we really love Julie's title, the Game of Drones project that she's got in the Scilly Isles, and we haven't been able to match that in its inventiveness and catchiness. But if we could, we might return to that later on. So the project is funded by the Seal Hayne Educational Trust and the Centre for Research Excellence in Intelligent and Sustainable Productive Systems, or CRISPS, at the University of Plymouth. And working on the project are Ollie, who we've just met, Professor Mary Knight, who many of you have met before at events like this held by B4, and myself. And what I'll do is I'll just give a brief overview about the background to the project and talk a little bit about the aims and objectives, and then I'd like to open it up to you to get your input on how we can get citizen scientists and beekeepers involved in our project. And I'll go quite quickly through the background because I'm sure many of you have heard similar things in the past. So just as a reminder, around Europe we have these main lineages of honeybees, the M lineage, the C lineage and A lineage, and I believe now the current interpretation is an out of Asia expansion of honeybees into Europe, and these ancient lineages evolved in the past, colonized Europe, the M lineage by a northern route, the C lineage by a southern route, and then Africa was colonized by an out of Asia expansion as well, but there's some A lineage influence in southern parts of Europe. And also owing to the glacial history of Europe over time, different subspecies and varieties evolved within these lineages as populations were left in refugia during the glacial cold period, and then they expanded out afterwards. And that led to the evolution of the honeybee diversity that we see today. The fact that beekeepers import bees a lot and move them around causes problems for this population structure that's evolved naturally over time. And I'll say a little bit more about that in a moment, but I think it's important to recognize that here we're talking about the loss of genetic integrity being a problem because it's moving species beyond the natural limits that they would move themselves, and humans are disturbing the natural population structure that's evolved, and that can then erode some of the diversity in the way it's structured spatially and geographically over time. So we've in the past been interested in levels of introgression, basically mixing of different genetic lineages between different populations of bumblebees, and you've seen many of these figures before, and of course Norman showed some earlier on today that were quite similar. And each line on these sorts of bar plots represents an individual bee, and the colours represent a different genetic signal. It's slightly badly arranged on this diagram, in that the green blocks here are both from Carneolum bees. The red block in the middle are from reference samples of Apis mellifera mellifera, and the blue block is from Apis mellifera ligustica Italian bees. And over here on this side we have samples from Cornwall that we sampled in 2018 and genotyped using microsatellite markers with Gabrielle Solland at Apigenics. And you can see when we've got a mix of colours in a single bar, those are showing admixed or introgressed bees with ancestry from the different lineages, the M and the C lineage. But we can see that we did see some relatively pure bees in the population that we sampled. We can also look at other sorts of analyses where we have clustering-based approaches, and here in this figure you can see that they're numbered in different ways. So we got 1, 4, 5, 3 and 2. So this cluster here, 2, 3 and 5, represents C lineage bees. This is a reference sample of M lineage bees, and these were bees that were sampled from Cornwall. And you can see on this plot, to interpret it and understand it, the points that are closer together are more genetically similar to one another. So we see our M cluster, our C cluster, and then in between are the bees that were sampled from Cornwall. So this again is some evidence that there's introgression between the M and the C lineage, but you can see some individual samples that were relatively close to the M cluster. So it goes to generally show that introgression is pretty widespread throughout the UK, but there are places where relatively pure colonies of AMM remain. And we followed up this work. So scientists that work in my kind of field, we use different sorts of genetic methods to do these analyses, and they've changed over time. There's been really rapid development of technology that's rapidly changed the scale of analyses that we can do, and nowadays we can use genomic methods as well as the methods that we used in the past. And some of you might remember Vic Buswell, a nice example I think of nominative determinism, that she worked on bees with a surname like Buswell. And she did whole genome sampling of honeybees around Britain and Ireland. And again, we've got one of these bar plots where, in this case, blue is representing M lineage bees, Carneca and Ligustica, and red is representing our Apis mellifera mellifera cluster. And these are samples from around Britain and Ireland. And again, you can see that there's quite extensive introgression, as you would expect, given the extent of imports that Norman spoke about earlier on today. But you can see, if you look on the figure, there are locations where there are relatively pure bees. And these also align with the results that Norman mentioned earlier on today, because Vic also found relatively pure bees in the Republic of Ireland, in Scotland and Colonsay, some close to Inverness, on the Isle of Man, and also on Jersey. So our past work is focused on managed colonies of bees, and we've often worked with beekeepers that like to conserve and keep AMM in particular, but this included some wider sampling. But what we'd like to do now is become more interested in free-living honeybees and do this sort of genetic work on free-living colonies to look at the level of introgression in the free-living population. And Ollie's obviously done his work in his PhD that we've just heard about on free-living colonies, and there's been work done in Ireland on these as well. Sorry, I've got ahead of myself. I forgot to say that, before I go on to that, the question is, well, why is this a problem, the introgression? And it's a problem because it breaks down local adaptation. So locally sourced bees will tend to outperform bees from other areas under their local conditions. That's because of evolution by natural selection, selecting for locally adapted variants. And in a sort of cliched phrase in population genetics, we talk about the evolution of co-adapted gene complexes, the assembly of lots of different genetic variants at different positions in the genome that lead to the evolution of local adaptation. Now, my analogy for this might not work really well, but I think one way to think about it, I know we've got Filipe here, who used to be a professional cyclist. Imagine if we wanted to have road cyclists go faster. If they wore an aero helmet, they might not go that much faster on with that alone, but if they had an aero helmet, tri-bars, 28mm tyres, deep section rims, an integrated headset, then they would now be on a really fast road cyclist. And so just as we could assemble a super fast road cyclist by having all of these different traits of a bike and a cyclist riding it, natural selection can assemble all of these traits about bees that they perform well locally. Now imagine into our population of road cyclists, we've suddenly introduced a load of mountain bikers. We've got 50mm knobbly tyres, baggy shorts and really wide handlebars, and then we're mixing up those traits, and the road cyclists wouldn't be performing as well anymore. And I think we can think about the loss of local adaptation in that way by analogy. We're mixing up traits from other sources that erode those that have evolved over time. And it's my analogy about having lots of different sort of sources of the local adaptation, and and my cycling analogy, the aero helmet, the wheels, the frame, and so on. Natural selection can act in that way as well. So it's not always acting on things that have a big effect, but on lots of small traits that on their own only have a little effect, but added up, summed up together, have a big effect and lead to the evolution of local adaptation. And that can happen when we have what you call polygenic selection. There are lots of genes that each have a small effect, but together they can contribute to the evolution of local adaptation. It can occur in other ways as well, but my point is that when you introduce individuals from other populations, then it introduces novel genetic variants, and that local adaptation that's been assembled by evolution, it becomes lost from the population. If you want a more concrete example than my cycling metaphor, there's a really famous case study in the Tatra mountain ibex. The Tatra mountain ibex was there's a rare and declining population of it in Slovakia and Poland, and to give the population a boost, you can often genetically rescue these populations by introducing individuals from elsewhere. They introduced ibex from the southeastern part of the range, and then the offspring of those matings, they rutted at the wrong time of year, which meant the kids were born in February, which is the coldest time of year in the Tatry, and then the whole population became extinct because the kids all died in the cold winters. So that's how you can get this loss of local adaptation, and that's why we're interested in detecting introgression in natural populations. But what we want to do is move on from our work on managed colonies and look at free living populations. And as I said, we've heard from Ollie in his PhD research. Some of you might be aware of research by Keith Brown and Grace McCormack in Ireland, where they also did survey work to look at the free living population, and they found them in various places like buildings, walls and trees and other places. And they also looked at the population genetically, and you can see that here we got in orange the C signal again from Ligustica and Carneca, and then in blue we've got the signal from Apis mellifera mellifera, and over here in Ireland the free living population was largely constituted of M lineage bees. But of course, we know that Ireland is a good reservoir of M lineage bees, and there's lots of M lineage bees in the managed populations. We don't know that it will be the case that the free living population in the UK will naturally be M lineage, and we might well detect a lot of C lineage introgression in free living honeybees in the UK because of the extent of gene flow from the C lineage inputs. Generally, in evolutionary biology, there's a lot of debate about when natural selection can maintain adaptation in the face of strong gene flow, and it gets quite complicated in terms of population genetic theory, but that's also of interest in this sort of study, whether or not we will see the maintenance of M lineage bees in the free living population, or we'll see a lot of introgression. And that's one of the things we want to do. And we're going to do that by finding colonies using the methods that Ollie's spoken about, sampling them, extracting DNA from the colonies, and then we genotype the colonies using molecular methods, and we're also going to investigate the morphometrics of the bees that we sample as well. But it's this first stage really that we're at in our project right now, is wanting to find colonies of free living honeybees. And from a logistic point of view, being based in Plymouth, we wanted to focus especially on the southwest of England initially, although we might extend further to other places in the south of England. And that's where we need citizen scientists, beekeepers, people interested in natural history to get on board with our survey. And I've a bit jokingly split this generationally. So if we think of millennials and Gen Z, we've got a website and we've got a QR code. You can scan that on your phone right now if you want to go through our website, and there's a reporting tool on the website. We could use things like Instagram, TikTok and X to spread the word about the project and get maybe younger generation involved in helping us search for bees. And then for boomers and Gen X, perhaps more traditional ways, engaging with beekeeper associations, natural history societies, village post offices and private estates. We did try Facebook. Funnily enough, if I speak to my students and I ask them about which sorts of social media they use, and I mentioned Facebook, they might say, oh my granddad uses Facebook. It's like, and we tried to make a Facebook site for the project, but the Facebook deleted the Facebook page, so we've lost it now. So we've decided to focus on the website instead. But what we really need is, we obviously want as many people out there searching for free living hives as possible. And this is where we want your input for a catchy title, input for engaging people, and how we can get a broader reach from the project. I'm basically really interested to hear your ideas about how we can recruit as many people as possible to search for free living colonies in the southwest. Oh yeah, and one thing I did want to add. I've been interested in insects since I was 13 years old. I'm sure any of you have been interested in insects for a long time. I think we forget how little the general public know about insects. So I worked with a master's student in the past who was from Guernsey, and she was conducting survey work for the Asian hornets on Guernsey, and the public had a reporting tool that they could use to report sightings of Asian hornets. But they didn't just report the things you'd expect them to get mixed up with Asian hornets, like social wasps, maybe other bees, and hoverflies and things like that. They reported burying beetles and clear-winged moths and all sorts of things that don't look anything like a hornet. So one of the trouble that we have is when we engage the public about recruiting them to look at insects, we're going to get perhaps like lots of records of tree bumblebees, Bombus hypnorum and things like that, and we don't want to end up wasting too much time searching for things that aren't honeybee colonies. So that is an issue that we have to balance into the project as well. So that is actually everything from me, and I'd just like to hear from you about how we can get you involved. And Matt. Yeah.
Well, I've just got a quick one. I did a similar kind of thing about 10 years ago where I lived to try and work out what's going on, and I actually gave rewards to young people. I gave them a fiver if they could find wild bees nests, and within a week I found about 15. Wow. So and that's, they're off on their bikes looking, and just interesting way of doing it.
Anybody else got a question?
There's one here.
Well, if nobody else has a question, it's just playing devil's advocate a little bit. So you just talk about the importance of preserving kind of local genetic pools that are adapted to local conditions. But, I think our environment is dynamic and is also changing. Are there not potential benefits to expanding your gene pool? I mean, climate change is a clear example. So, yeah. Yeah,
that's a good question. And I think you touched on something really important, that I think this project and others like it, they're not about treating nature as a sort of museum piece that we conserve as it was, but conservation biology at its best is about future-proofing populations and maintaining evolutionary processes. And then it does get quite interesting. So it is true that if you allowed mixing of the different honeybee populations and admixture, you would release a lot of genetic variance in the population, and that selection could act on those novel genetic variants that have been introduced. But at the same time, you would erode the sort of natural population structure that's evolved over time. And I think the best way to maintain the sort of future evolution of the population would be to maintain the natural population structure, but also maintain genetic diversity within those population fragments, and perhaps allow for some gene flow between the populations where it would naturally occur, and then that would maintain the genetic diversity and the structure of the genetic diversity, and that would be a good place to go from in terms of the future-proofing things. Yeah. But I think there could be other opinions where people would be less concerned about the loss of structure and more the maintenance of genetic variance in general. Yeah.